Hello everyone, I'm Steven and you're watching Steven Loves Science. And today we're going to learn about cell respiration, the process in which cells take glucose and convert it into a usable form of energy called ATP. So, the overall equation for cell respiration is shown here. C6H12O6, glucose, in the presence of oxygen, yields carbon dioxide, water, and energy, or ATP, which is the cell's energy currency. So, cell respiration can be broken down into three main stages. Glycolysis, which occurs in the cytoplasm, Krebs cycle, which occurs in the matrix of the mitochondria, and the electron transport chain, which occurs in the membrane of the mitochondria called the cristae. So, to get started with glycolysis. Glycolysis, what we're doing is, we're taking our 6-carbon sugar molecule and we're ripping it in half, and we're getting two 3-carbon sugar molecules known as pyruvate or pyruvic acid. We're also getting out 2 ATP to make glycolysis happen. We had to put in 2 ATP. We made 4 ATP. Now we have 2 left as a product. And what we're also doing is we're putting a passenger in a taxi. But what exactly does that mean? Taxis, electron taxis, or electron carriers are molecules that can accept electrons and hold on to them until they're needed. So basically what we're going to do is we're going to take energy from glucose and put it in a taxi, and that taxi is going to drive over to the electron transport chain, and that's what we're going to use to power the electron transport chain. So right now we're looking at a taxi called NAD+. That's when it does not have a passenger, when it's empty. When we put an electron to NAD+, we get NADH. You can think of H for high energy. NADH is a high energy molecule. Okay? So eventually we'll be able to drop off that electron at the electron transport chain, and we'll see how that works. Okay? So that's basically what we did in glycolysis. But remember, we also made this 3-carbon pyruvate molecule. So that pyruvate molecule is going to move into Krebs cycle. But before it moves into Krebs cycle, we do something to it. We rip off a carbon molecule that combines with oxygen, and we exhale it. So, remember the formula for cell respiration? CO2. That's where one of those carbon dioxide is coming from when we're initially ripping off our Krebs cycle, or we're initially ripping off our carbon here before it enters Krebs cycle to get a 2-carbon molecule, and then we change this, we do some chemistry to it, and we ultimately end up with something called acetyl-CoA. And that acetyl-CoA enters the Krebs cycle, goes through the Krebs cycle, we rip off another carbon, we get another carbon dioxide, combines with oxygen, and the Krebs cycle is then, again, going to put another passenger in our electron taxi. In the case of Krebs cycle, we're also making NADH, and we're making a close cousin of NADH called FADH2. Remember the H2, high energy, FAD plus is the oxidized form. Okay. All right. So, now we're ready to go into the electron transport chain. Our taxis are high in energy. They're really ready to give off their electrons in the electron transport chain. So remember we said earlier that the electron transport chain happens in the membrane of the mitochondria, the membrane of the cristae here. So let's go ahead and take a closer look at that. Okay, so here we have our electron carriers, NAD and FAD, and we're ready to give our electrons away. Our passengers have reached their destination. They're ready to leave the taxi. They have a long drive. They're really tired. They're ready to get out, and they're going to go through these proteins embedded in the membrane, and they're going to provide energy to pump hydrogen ions out of the membrane, out of the space here. They're pumping it out of the inner membrane space here. They're pumping it out. They're pumping it out. Okay? So what we're doing is we're going against diffusion. Remember, diffusion goes from an area of high concentration to an area of low concentration. What are we doing here? We're going from an area of low concentration to an area of high concentration. That requires energy. That's why we had to get our taxis filled with high energy passengers. Okay? So we're getting a lot of hydrogen ions here and not so many here. So this has a really positive charge compared to this. Okay, so now what have we done? We've actually created a situation that is now favorable for diffusion. We have an area of high concentration here, an area of low concentration here. So these hydrogen ions are going to want to go to the area of low concentration. The only way they can go to there is by moving through something called ATP synthase. And when the hydrogen ions move through that ATP synthase, they actually turn it, and using mechanical motion, the ATP synthase is able to tack on an inorganic phosphate group to an ADP, adenosine triphosphate, to make ATP, adenosine diphosphate, to make ATP, adenosine triphosphate. So that is called chemiosmosis. This process of pumping hydrogen ions out and then using the energy from the taxis and then allowing them to diffuse back in through the ATP synthase. Okay, that's all fine and dandy. So this is aerobic cell respiration. This is cell respiration that happens in the presence of oxygen. But let's say I'm Usain Bolt. I'm a runner. I'm running so fast and my muscles, my myocytes, my muscles are requiring more energy than we can get to them during the process of aerobic cell respiration. Going through Krebs cycle, doing electron transport chain, simply takes too long. These myocyte cells need energy now. They need to get Usain Bolt to the finish line. So what are they going to do? They're only going to do glycolysis. Okay, they're only going to do glycolysis. But what happens by only doing glycolysis? Remember the products of glycolysis? We got NADH and pyruvate. But remember in the electron transport chain, we ended up ripping the H off of the NADH. So we were kept NAD+, and it could kept being recycled through the process. But now we have a buildup of NADH, and we eventually run out of NAD. So we can no longer do glycolysis. So we're stuck. So luckily, the body has evolved in a genius way, cells have evolved in a genius way to combat this process. And that's called lactic acid fermentation. So since we have this buildup of NADH, we have to get rid of the H in order for it to continue to happen. So what we do is we convert pyruvate into something called lactic acid, and that lactic acid is then able to accept the electron from NADH. So it's able to act as an acceptor of the electron. And what happens is lactic acid is a poison, and it actually builds up in your muscle cells, it builds up in your myocytes, and that's why you feel a burn after doing strenuous exercise, because the lactic acid buildup is actually a poison, is killing the cells. Okay? So once you finish running and you're breathing, you're actually degrading the lactic acid, and that's going away, and you're degrading the lactic acid, and that's why you no longer feel a burn. All right, so thanks for watching this episode of Steven Loves Science. I hope this aided in your understanding of cell respiration. Sorry it was a little all over the place today, just getting back in the groove of making videos after a long break. Please stick with Steven Loves Science. We have some really cool stuff planned for you this summer. We're gonna be watching a pediatric open heart surgery. We're going to be going to Life Technology headquarters in Carlsbad, California, and San Diego, and just a bunch of other cool stuff planned for this third installment of Steven Loves Science. Please stick with us. Thank you for watching. Please subscribe, rate, and comment.